Hello, everyone. My name is Renee Long, a communications specialist and webinar coordinator here at the American College of Healthcare Sciences. And joining me today behind the scenes for all our technical needs is Dominic Aiello, a social media specialist and webinar coordinator here at ACHS. And it's my pleasure to welcome you to today's Master Lecture webinar, Are Your Genes Depressing You? MTHFR and Your Mood, presented by functional nutritionist and master nutrition therapist Coleen Walsh. So just a few housekeeping items before we get started. You may have noticed that your line has been muted, and we are recording today's webinar, so this helps ensure we can clearly hear our presenter. And you'll also notice that you have a control panel at the right-hand side of your screen. If you have a question for Coleen during the webinar, go ahead and type it into the questions box, and I will read and answer them during the and Coleen will answer them during the 10-minute Q&A session at the end of the webinar. And if you have further questions that require a bit more of in-depth answer, please go ahead and follow up with Coleen directly at info@harvestingnutrition.com, and you'll see that email address at the end of the webinar. And she's happy to respond to all of your questions, but just please be sure to give her some time to get back to you. And now I'll go ahead and turn the webinar over to Coleen, who will give a brief introduction and then begin her lecture. So welcome, Coleen. You should now have control of the screen.
Great. Thank you so much, Renee. It's so good to be here, and I appreciate all your time and Dominic's time and help in asking me to do this webinar today. I hope everyone will really enjoy it and get some good information and understanding about MTHFR and methylation. Just a quick note for myself that this is just a disclaimer that you can read over. This is not to provide any medical advice, to diagnose, or to treat. This is for informational and educational purposes only, and it's not any substitute at all for any professional medical advice, because we're all unique individuals that everyone needs individual attention. Just a little bit about myself. I'm a functional nutritionist and a master nutrition therapist. I'm called the Methyl Queen because of my work in MTHFR and methylation. And I'm a professor, educator, and speaker. I have a forthcoming book, Thriving with MTHFR, a guide for the everyday mutant. And I was the National Association of Nutrition Professionals 2014 Impact Award winner last year for this work. So I'm very excited. I'm very passionate about it. I am not a biochemist or a scientist, but I am an everyday mutant, as I call myself, and I got very interested in this subject because I really was very sick and I wanted to take charge of my health. And so I began to research and study more and more and more, and now I'm very happy to be teaching this subject. So for today, we'll be reviewing the big picture of what MTHFR is and the whole big picture of methylation, because there's so many people out there that just go into MTHFR and it's so much of a bigger picture. I'll tell you about 60 to 75% of my clients come to me because they were treated by another practitioner or physician, and they just looked at MTHFR alone. But we have a lot of other mutations in these cycles that could come into play, and we're also individual that we shouldn't be going over just one specific recommendation or supplementation or even prescription. We'll review health consequences, and we'll talk about B vitamins, how they act as cofactors in these pathways. Also, we'll get in a little bit more detail with the BH4 cycle and talk a little bit more about mood and psychological disorders. We'll just finish with some take-home notes, and I'll answer some questions that you might have about this topic. So I don't know if you have studied this or not yet, but this is Dr. Ben Lynch's from seekinghealth.org pathway planner. I've been given permission to show this. This is a pretty complicated pathway. Today, this is another one by Dr. Amy Yasko, but today we'll start with my methylation map, and this is just a very basic understanding of those complicated maps that you just saw. So we'll look at the big picture. This is my simplified version. This is the methylation cycle, which has evolved really over the years. It started just as this right orange methionine cycle, and that's what they called the methylation cycle, along with a transsulfuration pathway. And Dr. Amy Yasko kind of added these, and different researchers added these together because they saw how many nutrients are really combined and really passed on and connect in these pathways that are put together. So we'll be discussing this. But you can see that MTHFR is right in the center. It's at a very crucial point in these pathways, and we'll discuss this just a little bit today. And that's why a lot of people really focus on MTHFR. But my trainings are like a 101 or 201 to Dr. Yasko's and Dr. Lynch's deep science and research. So I really break these pathways down. And today you'll get a little taste of that. So this is, again, we all know what this looks like. It's the Olympic rings. And I really talk about the methylation cycle in different ways. And one of them is how the Olympic rings are all independent, single individual circles, but they come together and they make a bigger function, and they see that. So just as that happens, we have the methylation cycle. So each one of these circles, or the line at the bottom, is individual, but when together they combine, they have a bigger meaning. So they work independently, but they also work individually. And this all happens in our cell. So when we get cellular, we'll just talk a little bit about this very quickly. But a repeat, if you've taken your biochemistry classes, in each cell, or even 5th grade biology, you probably might remember some of this, every cell in our body has a nucleus. And in that nucleus, we have our chromosomes. The chromosome is organized into 23 pairs, and it contains this double helix of the DNA. And our DNA is in each and every cell of our body. It is divided into segments called genes, and we'll be discussing these genes today. And we have 2 copies of each gene, or what we call these nucleotide or bases, these nucleotide bases. And so today, when we talk about MTHFR, we're talking about, I'm going to go back one more time, we're talking about these specific genes. So our genes have the instructions to create proteins in our body. And they do that by taking the amino acids that we get from food and putting them together with these instructions, or maybe cookbook, right? And guided cookbook and instructions that the genes tell us to line up these amino acids in a certain way. And those proteins act as different things in our body, from hormones to muscles, and also what we call enzymes that perform metabolic reactions in biochemical pathways. And so these genes can have certain SNPs, or single nucleotide polymorphisms. So that nucleotide is that base of the DNA. And so we just call them SNPs for shorts. And SNPs are permanent errors in our DNA. So we kind of talk about them as a typo, right? So instead of a C being right here, it actually turns into a G. The same thing could be with one of the A's or the T's. Instead of having those 2 exact pairs that are identical, one of them is switched. So there's like a typo or a permanent error. And that relates to a single base difference in that nucleotide. We inherit these through our biological parents. So each parent has 2 of each gene, and they give us 1 copy of each. And so we get 1 from our mother and 1 from our father. And it's the most common genetic variation, these SNPs. We have about 10 million SNPs in the human genome. They really show us the differences among people. Some of these can be our hair color, our eye color, and others can even be like personality traits. So one common statement is that most SNPs have no effect on our health or developments. And I put several question marks there because I believe we are still learning so much about our genetics, and in particularly our epigenetics, what lies on top, and how our environment and diet and lifestyle factors trigger our genetics to turn on or turn off. That I put question marks there. They used to call junk DNA. But now we're knowing, after they research more and more and start learning about what they call junk DNA, this DNA has so many things that it is involved in, and so they really don't think there's any junk DNA. We're just not up to that capability of understanding what is happening. So these SNPs, and my computer's just a little slow. Oops, sorry about that. These SNPs can happen in the methylation cycle. And we'll just talk a little bit about the methylation. And MTHFR is one of those SNPs. So it's a change in the nucleotide base. And we can have one SNP or we can have two SNPs. And that means we have either one which is homozygous or one which is heterozygous or two which is homozygous. And those SNPs can be considered predisposing us to diseases. Usually it's just not one SNP that does that, but a combination of SNPs. And MTHFR is one SNP. The methylation cycle, so this whole little map here to the right, includes 5 major biochemical cycles in our body. It really, the most basic job of this methylation cycle is to take nutrients that we obtain from food and change them into different molecules to make them more bioavailable or usable in our body. And the functional status of this methylation cycle, or how well it's working, really will determine our resistance or susceptibility to environmental toxins and microbes and really disease. But before we go into details, we'll just understand these 5 pathways. So the yellow is the urea cycle, and this mostly happens in the kidneys and detoxifying ammonia and our liver as well. The BH4 cycle, which we'll get a little bit more into detail, which is in purple here. That is our neurotransmitter cycle, which people call. That's how we process amino acids, tryptophan and tyrosine, or phenylalanine beforehand, into our neurotransmitters, dopamine and serotonin, and then into hormones as well, and degradation of hormones. We'll be going a little bit more deeper into the BH4 cycle. The blue is the folate cycle, and that's how we process the certain nutrient folates from foods. Just a quick note on folate and folic acid. Folic acid is the synthetic form, and it actually is processed a little bit differently in here. So when we eat folate from foods, it has different versions of folate. The most bioavailable form is methyl tetrahydrofolate, or MTHF. And that MTHFR enzyme is that last step. It speeds up that chemical reaction from the instructions from our DNA, and it speeds up the chemical reaction to create this methylfolate. And this is the most biological and bioavailable form of folate in our bodies. But there's several different forms above here. Folic acid would not be considered one of the folates because it's the synthetic form. So the folic acid would be up here. And then on the right-hand side, we have the orange, which is the methionine cycle, and that processes the amino acid methionine and down to homocysteine and then back around. It creates great nutrients like SAM and energy, and goes to protect our DNA. Homocysteine down is the transfer sulfuration pathway, and that creates glutathione, and it helps to detoxify ammonia from our bodies. It helps to create ammonia, which is then detoxified through the urea cycle. So this is kind of the whole methylation cycle that we'll be talking about today. It manages or contributes to crucial functions that include things like detoxification, our immune function, our energy production. It helps us balance our moods and controls our inflammation. So it also really contributes lots of nutrients like CoQ10 and creatinine and glutathione, our master antioxidant and master detoxifier. And it processes all of these neurotransmitters to help us have those mood balancing and feel good symptoms rather than anxiety and mood disorders. And it also gives energy, like I said, CoQ10 and creatinine and carnitine. It also protects our DNA. And this methyl group right here, and this is kind of the baby that they call, why they call the methylation cycle, is this starts off right after MTHFR. After MTHFR, that enzyme creates this methyl group that's attached to a folate, so that's methyl tetrahydrofolate, that last step in the folate pathway. And this methyl group then kind of moves on and is passed on to other nutrients in the methionine cycle to the right. And this methyl group, or methyl tag, is very important. It's a very important nutrient and needed in our bodies. So there's certain health consequences when we have these certain SNPs like MTHFR. Again, I'll just say that it's not only MTHFR, it's usually a combined effect with MTHFR, other SNPs in these pathways, as well as epigenetic factors like our diet and lifestyle and how we manage stress and how we think about stress. But the four big ones I call the big four. Most scientific studies are really focused on these with MTHFR and other methylation cycle SNPs. And one is cardiovascular disease, which is very important due to inflammation, due to homocysteine rising, oxidative stress, etc. Another important one you might know of is pregnancy, fertility, and birth defects. A lot of miscarriages will happen with people with MTHFR, birth defects and neural tube defects, spina bifida, and fertility problems. So that's another heavily studied area. And then a third one would be cancer, especially estrogen-dominant cancers, because this BH4 cycle has to do with actually degrading our estrogen and metabolizing it. And then our last big four is mood and psychiatric disorders, which we'll get into a little bit more. There's a lot of other ones. The close fifth one, I would say, is thyroid dysfunction. There's a lot of connection with that, and MTHFR and methylation. And then other associated risks are autoimmune diseases and chronic viral infections like Lyme and Epstein-Barr. And a lot of this is due to the overall lowered immunity and the inability to detoxify due to these MTHFR and methylation SNPs. So you can see there's a lot more. This is just a combined correlation and scientific studies that are shown. But it basically all breaks down to the inability to process nutrients that lead to nutrient deficiencies in our cells. And as we know, as I said earlier, that the methylation cycle really just processes nutrients that we get from food and breaks them down into more bioavailable forms that our body uses in our cells and our body needs, especially folate and B12 and our neurotransmitters. So there's lots of nutrients that are passed back and forth, like that methyl group, that if this methylation cycle is slowed down, it could lead to these associated diseases and risks. And so we'll just delve deeper a little bit why mood and psychiatric disorders are one of them. Some of those would be depression. Schizophrenia is a big one, especially with MTHFR and some other SNPs like COMT and MAOA and B. Bipolar disorder. Addictions are very common because we need those B vitamins to really sustain our mood and our neurotransmitters and our hormones. And if we're not stabilizing all of those things, then addictive patterns can start to happen. Anxiety is another big one, especially when your neurotransmitters are not balanced correctly. ADD, ADHD, OCD, autism is very connected, dementia and Alzheimer's disease. Alzheimer's disease is very connected as well. And I just pulled a couple of these studies just to quickly show. And this was, I think, a meta-analysis that came out, and it was just showing the connection between mood disorders and MTHFR, what we call compound heterozygous, which is one variant of the 677 and one variant of the 1298. So the heterozygous meaning you have just one of each. And then they also did the homozygous ones. So, and they said the common psychiatric disorders, depression, anxiety disorders, bipolar, schizophrenia are connected. Just another one showing the 677 polymorphism in children and adults. And the homocysteine that I mentioned. Because with 677 MTHFR, the homocysteine can be raised. And studies have shown that with proper version of methyl, folate, and B12, and I know this says folic acid, but other studies have shown that folate and methyl folate in particular and B12, in particularly methyl B12, it can lower our homocysteine, taking down cardiovascular rates and reducing some of the mood disorders as well. And just a last one, just to show you kind of a little bit of what's out there. B vitamin polymorphisms in behavior, evidence of associated with neurodevelopment, depression, schizophrenia, bipolar disorder, and cognitive decline. So again, it says folic acid here, but folate, B12, and B6 are essential for our function of our brain. And I'll tell you again the difference. Sometimes, and this is a newer study, most of the newer studies actually are showing the different versions of folate when they said that, when they actually do. But unfortunately, some use the term folic acid as all folates instead of the certain folates. So normally the newer ones will specify what folate they're talking about because we know folic acid is a synthetic supplement and it's not natural to our body, so our body doesn't actually naturally produce it. Okay, so just B vitamins here. So as you just saw, there's a lot of B vitamins, and in these pathways, they're used as cofactors. And so each one of these pathways has several biochemical steps. And each one of these steps needs an enzyme, like the MTHFR enzyme, and then there's other enzymes for each one of these different steps. So like the MTHFR enzyme, we need the enzyme, we need cofactors and coenzymes, and we need a substrate or another product to kind of take that to the next step and to make an end product. So with the MTHFR, we have a substrate, which is the 5,10 methylene tetrahydrofolate. And then we need the MTHFR enzyme to come in. We need some precursors to niacin, B vitamins, to be the coenzyme. And then we go on to the step to create actually the methylfolate, the methyl version of folate. So each one of these steps requires B vitamins, and very important B vitamins. Of course, we just talked about folate. And B12 is very important as well in these pathways. Precursors of biotin and niacin are through there, and also B2, which is the riboflavin. So B vitamins are pretty important in these pathways. As we know, they're important for our brain function and for our neurons to actually work correctly. So again, the folic acid, I know it does say it everywhere. That just is a common term that everyone started using. But the more appropriate term is folates when we're talking about the actual vitamins that our body creates, or the vitamins that are in naturally in food. So here is my methylation map again. We'll go just a little bit more into detail, and you can see how they spin together. I call it like cogs or wheels. So if you were to look at a watch or an old clock and you take off the face, you can see these cogs or wheels keep spinning. And if one slows down, the other ones are going to slow down. So these just keep pushing nutrients and sharing nutrients to each other. Again, this is just a very simplified version of this map because it gets very confusing. But we'll just look again. We have the urea cycle that actually helps to detoxify ammonia and other nutrients. And then the BH4, or another referred to the neurotransmitter cycle. And that really processes the amino acids into serotonin and dopamine, and then down onto from dopamine into norepinephrine and epinephrine, serotonin into melatonin. But in order for that to happen, we need BH4, and BH4 is created from BH2 and methylfolate. So methylfolate, that B vitamin, acts as kind of the cofactor to create this BH4. And then BH4 is needed for tryptophan and tyrosine to do their job, making the serotonin and the dopamine. So you can see how MTHFR is important for the BH4 cycle. Not only does it help with the BH4, it also is helping process the tyrosine and the tryptophan to create these nutrients below. And we talked about homocysteine a second ago. So this methylfolate and the one carbon metabolism, or that methyl donor. So the MTHFR enzyme before this is not the full three hydrogens in one carbon, right? It is 2 hydrogens in 1 carbon. After this happens, it adds a hydrogen to make the methyl version of folate. And that methylfolate comes together with homocysteine. The methyl group gives to homocysteine to create methionine to keep spinning around. And then that methyl group that leaves turns right back into the tetrahydrofolate to keep this one spinning. So this continues to spin, continues to create this methyl tetrahydrofolate, and that methyl group brings in with homocysteine, gives that methyl group to homocysteine, that changes to methionine. Also B12 is a cofactor right there. And also that methyl group comes over here, that methylfolate comes over here and helps to create BH4. So you can see kind of how MTHFR is in the center. We'll talk a little bit about BH4. So the BH4 cycle, it's really the neurotransmitter production from our proteins that we get from our diet. It assists to break down phenylalanine into tyrosine, back into dopamine, and then to noradrenaline or adrenaline, or norepinephrine and epinephrine. And you can see on the right, and I know it says folic acid. I probably should have just crossed that out and write folate on this, but this really kind of shows that each step is involved. You have all of these cofactors, which usually are B vitamins, and coenzymes, which are usually magnesium and zinc and different other elemental minerals. Thank you. And along with this, you need the enzyme to actually speed up this chemical reaction. So each one of these enzymes would be a different and have a different name. And those, again, those enzymes are created from amino acids with the instructions by our DNA. So a cofactor to, it's also the BH4 is a cofactor to produce nitric oxide, which is really important for our cardiovascular system, for our endothelial cells to be a vasodilator, to help dilate our blood vessels, so the oxygen can really move through. It starts with the cofactor of methyl tetrahydrofolate, right? So that methylfolate. BH4 plays a direct role in the urea cycle through the MTHFR and through the BH4 cycle. If there's a low BH4, there's different toxic metabolites that will actually be generated or created because we don't have enough BH4. And so one of the things that BH4 does is detoxifies ammonia in this urea cycle. And we need 2 molecules of BH4 to detoxify every 1 molecule of ammonia. And so if we don't have enough, superoxide will be generated. And that's almost a free radical that can happen. Another free radical is peroxynitrate, that if we don't have enough BH4, it will be generated. So BH4 is a really important thing, not only because we're making these great neurotransmitters, but it helps to detoxify our ammonia and keep this urea cycle important. And methylfolate actually recycles the BH4. So it's really necessary to keep this cycle going. So here's just an equation: low MTHF, or low methyl tetrahydrofolate, that last step of the proper folate form, equals low BH4. And if we have low BH4, that will equal lowered ability to create neurotransmitters. And here's just another photo that we can see. The tryptophan goes into 5-HTP. That 5-HTP goes to create serotonin. And that serotonin actually even goes down to create melatonin. So again, this is just a very simplified version of these pathways. They get very complicated once you get a little deeper into them. On the right side, the phenylalanine goes into tyrosine, goes into, there's a couple other steps that aren't here, L-DOPA, and goes into dopamine, and this is another step to go into norepinephrine or noradrenaline, and epinephrine or adrenaline. And so these are extra steps. Again, B vitamins are one of the major cofactors. So it's really important to have our B vitamins not only from our food, but to have a really good gut function so that we're able to digest them properly, absorb them properly. And other cofactors, C and zinc, TMG, which is trimethylglycine, and SAMe, S-adenylmethionine, which is the biggest methyl donor in our body. That is created from the amino acid methionine, but more so methylfolate. So methylfolate gives that methyl group to homocysteine to create methionine. Methionine creates SAM. So these are really important. Other things to improve our neurotransmitter reception is omega-3 fatty acids. So nutrients, of course, are the roots of all of our ill effects if we're not getting the nutrients that our body needs to create these biochemical pathways to create the substances we need to think like our neurotransmitters, then we're going to be slowed down with our moods. And just another way to see this. Again, there's a couple more steps here. You'll see some of these are enzymes that help catalyze and speed up these chemical reactions. Here, tryptophan down to serotonin to melatonin, and then phenylalanine to tyrosine down to dopamine, norepinephrine, and epinephrine. Two other SNPs are MAO and COMT, actually MAOB, this one is, and COMT that kind of play off of the tyrosine to dopamine and or down to norepinephrine. As well as there's an MAOA over here that plays right between serotonin and melatonin. So you can see just another version of this geeky stuff. So, oops, let me back up one more time. So the BH4 cycle is pretty crucial in to actually creating these nutrients. You'll see that the folate cycle, again, breaks down. The folic acid is up here. It's the non-synthetic form, but it breaks down to several different steps before we hit the MTHFR enzyme that catalyzes and pushes that reaction to create that methylfolate. That methylfolate goes over here to tag that methyl group to homocysteine, and homocysteine with that methyl group turns into methionine. B12 is involved right there as well to turn into methionine. And then that continues to spin, and that one important nutrient, SAM, and SAM comes over here to help some of those neurotransmitters as well, and donates that methyl group for energy and DNA protection. It's one of the biggest methyl donors, and it's a very important nutrient. So although methionine from our diet can start to create that, methylfolate really creates that as well. And then on the other side, this methylfolate actually helps to create this BH4. And this BH4 is needed to create, use tryptophan and tyrosine to create our serotonin and our dopamine and down that pathway. So it's all combined, and MTHFR is an important point. But again, there's a lot of different SNPs that we have here. Each one of these steps requires SNPs, and there's a lot of steps right here. Not requires SNPs, I'm sorry, requires enzymes that can be SNPed. And then we need the proper BH4 to actually detoxify this ammonia. And so we need 2 BH4s to detoxify every 1 molecule of ammonia. And if not, those free radicals can be actually created if we don't have enough. So just another version, you can kind of see this moving around and how kind of these nutrients react to each other. A very simplified version of these pathways. So a lot of nutrients are passed back and forth. Okay. Oops. I'm sorry. There was one slide missing right here. Just an important note, I wanted you guys to know that not all, when I say SNPs, so I'm talking about the SNPs, meaning that there is 1 or 2 nucleotide bases that is changed, or those typos that happened. You could totally be perfectly normal, or what is called a wild type with these. So that means that there is there's no error or mutation involved. So when I'm talking about SNPs, it means when people do have them. But just the presence of a SNP doesn't mean that there will be a problem, that someone's going to be sick or have some deficiency in the nutrients. But the absence of a SNP does not mean that the pathway is working properly either, right? Because we talked about these epigenetic factors that can happen. So just because you have these genes doesn't mean you're going to be sick. And I constantly need to remind my clients that, because there's so many clients that call me very nervous. Oh my gosh, I just got this report. But what I tell them is that our genes load the gun. But the environment, like our diet, how we perceive and handle stress, how we sleep, how we exercise, all of these lifestyle factors, and our toxic environment, all of these things are the environment, and our environment really pulls the trigger. So it's not genes alone, and up to even 75% of that can be determined. How our genes are reacting can be determined how we live our life and how we eat our food. So just having that SNP does not mean that there's a problem, but also the absence of a SNP doesn't mean that the pathway can be working properly. If we don't have these nutrients that are important as cofactors, or that are important to create these enzymes, then the pathway could not be working properly as well. Stress interferes with these pathways. Heavy metals interferes with these pathways. So the idea of epigenetics and how this expressing of the gene or expressing of this SNP may or may not be happening. So it basically goes down to, again, decreased ability to convert nutrients to usable forms in our body. And that will be dependent on each person and their individual, not only genetics, but their SNPs as well. So a quick note on that, talking about epigenetics, is this is a great quote from a book called 5 to Thrive. It is actually now been renamed The Definitive Guide to Cancer. And I loved it, the quote that it said, "We cannot change inherited genetic mutations. We can, however, utilize our lifestyle to minimize exposure to further mutations and, importantly, to alter how our genes behave. Thus, to understand how to alter our epigenome is to understand how to best prevent cancer." And so since this was a cancer guide, it says cancer, but I would add to that, best prevent cancer and other diseases. And so there is great, a good outlook for that. And then I just have a couple how to reach me. My Facebook is Harvesting Nutrition. My Twitter is Methyl Queen. I have lots of great things on Pinterest if you do that. And as Renee said earlier, info@harvestingnutrition.com is my email address. If you go to harvestingnutrition.com, I have a free MTHFR Quick Start Guide. That and newsletter. If you fill in your name and your email, and it just gives some information on genetic testing. And don't worry if in the top 5 things you should know first if you find out about MTHFR or other SNPs. Also, I have Thriving with MTHFR, a guide for the everyday mutant, practitioner education videos. I also have videos for the everyday mutant. It's a 3-hour training program, and a forthcoming book and guided program later this year. So that is the end of my presentation, and we can go into Q&A now. So I'll pass it over to Renee.
Awesome. Thank you, Coleen. That was an excellent presentation, and we already have some questions up in our chat box here, so I'll just go ahead and start with the first one. So we have a question from Kim, and she says, or she would like to know, you mentioned that treating MTHFR alone can be problematic. Where do you start with treatment?
Yeah, that is, it's a very detailed process, and I'll tell you that I actually spend 8 to 10 hours with my new clients because it's so complicated. And I know I touched upon it a little bit, but I really look into each individual's, not only their genetics, but their history, their symptoms, their lab tests, and their toxic load, their GI health. And I kind of put all of these pieces of the puzzle together within the genetic portion to determine which supplements would be necessary for them. So, like for example, if someone has, we did mention COMT and MAOA and B. If they have those and MTHFR, you know, some people may not, they might have problems, and that's expressing and that's causing a problem. Some of those people might not be able to do the proper form, like the methylfolate or the methylcobalamin. They might have to do like folinic acid and do maybe a different form of the B12, like hydroxycobalamin. So it is a long process that's individual for each and every person. And it gets very complicated with that. So what I do in my 5-hour training is I really talk to nutritionists and other healthcare practitioners, everyone from naturopathic doctors to MDs to chiropractors, and I really teach them how to start with diet and lifestyle, and then really be careful with detoxification and in certain protocols with nutrients, because we can start there first with our diet, with our lifestyle practices, with our toxic load, and starting small, because people can really have bad reactions to supplementation and detoxification protocols. So diet and lifestyle are always cleaning up that, and cleaning up our environment is always number one. And stress reduction is another big thing.
Great. So our next question comes from Suzanne, and she would like to know, what does BH4 stand for?
It's tetrahydrobiopterin. So it's basically 4 molecules of biopterin. Mm-hmm. And the BH2 is 2 hydrogen molecules and biopterin, sorry, and 4 hydrogen molecules and biopterin together. So tetrahydrobiopterin.
Great. So hopefully that was helpful to you, Suzanne. So our next question comes from Sarah, and she says, I have heard that folic acid is the lab-created or synthetic form of folate and is not preferable for supplementation. In addition, any supplement containing folic acid should be avoided, and one that uses folate should be used instead. Do you agree?
Absolutely. I think, I hope I said that a couple times in the presentation. Mm-hmm. This question might have been before we talked about that, but absolutely. So it's a synthetic form, and there's different forms of folates, and depending on your genetics, depending on your symptoms, your history, your family history, etc., the different forms of folate would be best for you, and that's kind of individual per person.
Great. Yeah, and I think you did mention that a few times, so that info should be in the slides as well. So our next question comes from Carissa, and she would like to know, are there other ways to test for other SNPs like MAO and COMT? And also, what is the one best way to correct this lack of methylation process?
Okay, thank you, Carissa, for your question. I'll answer that last part first because there's no answer for that. Sorry, there's no one best way. It's just if your methylation is kind of tweaked, it's going to be a little bit of a long process, and you need to work with someone that's really knowledgeable in not only MTHFR but other SNPs, especially if you have MAOA or COMT or some of these other SNPs that can especially come into play since we're talking about depression and mood disorders, psychiatric disorders. One of the best ways to test, and the cheapest ways, is 23andMe. I don't necessarily like their interpretation, and some of you may know that the FDA stopped some of their interpretation last year, but they're working with them now. But what you can do is you can go to 23andMe. It's $99. I have the link and I have information on how to do this in my free MTHFR Quick Start Guide if you go to my website. And so it's the cheapest, it's the most accessible, and it's the most affordable form. And then you take it to different sites, and I talk on the Quick Start Guide for that as well. You can take it to other sites, and then you get that information interpreted. So then it just kind of comes with a report of kind of yellow, green, or red, and it tells you which ones you're green or wild type or completely normal, the yellow, which are heterozygous, and the red, which are homozygous if you have two of those. And it will just kind of spit out, it will say MTHFR, homozygous plus plus, or COMT. And so it's a nice read, and it's easy to kind of see which SNPs you do have and what you're working with. So, but yeah, I think I have some good information in that free quick start guide that you can get better. There's a lot of other testing that you can do, but it usually requires either a prescription from your doctor, or expensive. And just some of the MTHFR tests alone are like $395. And then some of these other tests go up to $1,000 to look at more methylation SNPs. So, and I think actually, I don't know if you're still on mine. Oh, good. Sorry. I have this last slide that kind of shows a whole bunch of these SNPs. So this is another one of Dr. Amy Yasko's pathway maps. And so all of these colored circles are different SNPs. There's a whole bunch more, but a lot of the tests, the 23andMe will test, and we can they'll show you and spit out the results for these. So I think it's a really accessible and affordable way.
Great. So our next question comes from Kim, and she would like to know, but if you have the mutations, will aging alone trigger their expression?
You know, it just depends on that, Kim. I'm sorry, there's no yes or no. It's it's a very complicated pathways. It's very complicated. Epigenetics is, and it's all independent on each person. It usually does get worse or express a little bit more with age, because with age we have increased oxidation, we have increased free radical damage, we have increased inflammation, and so all of these things can turn on to express a little bit more as we age. Plus we have our toxic load, and then those diets and the alcohol and all of those other things start to kind of add up as we age. So some of the things that we weren't so good with ourselves or with our body can increase all of these problems that will turn these SNPs to expression.
Great. And so our next question is from Carissa, and she would like to know, can you over-methylate?
Over-methylate? Yes. You can definitely over-methylate, and that's why I kind of give a warning to that not everyone should be taking methylfolate and methylcobalamin. As I said, I think like 65 to 75% of my clients come to me because someone prescribed or gave them high-dose methylcobalamin and methylfolate, and actually they had very severe problems. Some of them, like one of my clients even felt that they wanted to go into a insane asylum. They were like, I think I needed to go in to be admitted to a psychiatric ward.
Oh, no.
Because of the methyl versions. Oh. And so that's kind of what they call referred to as over-methylation by supplementation. And so it's really important especially if you have any mood or psychiatric disorders, not to just push these nutrients when you don't know what's happening, because they're pretty involved nutrients, and they're pretty powerful. They've changed my life and my health in a great way. But I've seen working with not the correct supplementation per individual, it could be devastating, and that's not going to happen long term. It will just be probably for a week to several weeks, or even a day. So yes, there's definitely ways to over-methylate.
Great. Yeah, that's definitely something to keep in mind for sure. And so Suzanne would also like to know, is there a test for MTHFR?
Yes, and we just talked about that.
Right.
You usually, like the top four, the big four, what I say, the fertility, the cardiovascular disease, the cancers, and the the mood disorders and psychiatric disorders. Most doctors who specialize in those, MDs, they're learning and they're understanding about MTHFR. They might not have the full picture, but they'll, if you have those and if they run in the family, those doctors might order a test and test for MTHFR only. And that's when people, some of my clients come to me with just the MTHFR, and then I have them go to and get the full panel with the methylation profile with the 23andMe.
Great. Sorry, scrolling to find these questions here. Okay, so our next question is from Beth, and she would, her question is, can you speak specifically to how this cycle works to convert, and I'm going to butcher these, cyanocobalamin to methylcobalamin, am I saying that right, etc., to be usable in the body? I'm curious what if the involvement is with this regarding BH4 and BH2, etc.
It doesn't necessarily involve BH4 and BH2 with the B12. Most of the B12 is the methyl version of cobalamin, and that in the methylation cycle is the methyl version. The conversion of the cyanocobalamin and methylcobalamin actually happens a little bit outside this pathway, but in the cell. So this usually takes the methylcobalamin. The problem with this is just someone getting too many methyl donors if they already have methylation. So different versions of B12 would be used, not necessarily for this exact point, but to increase your B12 so that you can create methyl B12. So it gets a little bit more confusing. It doesn't necessarily have to do with the BH4 cycle. It's more about, actually you can see these two arrows that I put out here, the methylfolate connecting with the homocysteine and then coming around B12 as a cofactor, and this MTR helps to, that enzyme helps to catalyze that reaction, and then the MTR recycles that B12. So just that's a kind of a brief answer to a more complicated, which I don't think we'll have time to talk about. So I hope that helps, Beth.
Right. Right. And Beth, you can always follow up with Coleen too at info@harvestingnutrition.com, or just visit her website, and you can get in touch with her there if you had any more in-depth questions. And so Chris would like to know, how do you find someone knowledgeable in this area? And I'm wondering, Chris, if you maybe meet like an MD or an ND, because obviously Coleen is very knowledgeable in this area, so you can always reach out to her. But Coleen, I didn't know if you had any other thoughts on that question.
Sure. Yeah, I'm listed with Dr. Lynch, his MTHFR.net. He lists different doctors in different places. I would say from that list, there's other places that do Dr. Amy Yasko's work a little bit more, and she deals a lot with autistic population and does things a little bit differently than Dr. Lynch. But I would start with MTHFR.net, and they have a listing of a lot of doctors. And then his listing, he doesn't vet anyone. So his listing is people who have taken one of his 2-day trainings. And they're very involved, and I know a lot, and that's why I did my 5-hour practitioner training program, is because I went to that, I understood, but so many people didn't understand what he was talking about because they didn't have that basic or that base understanding, and he was going pretty heavily involved in those workshops. So what I would do is, if you're taking a doctor from that list, I would suggest you to call them and start asking them questions and asking if they deal with these other SNPs, or is it just MTHFR, and if they have a better understanding of this whole thing. Because I think people would just go to that one thing. They were hoping to get kind of a packaged deal of how to treat people. The MDs were, but as you guys kind of see just from just a quick version of today, it's really involved, and there's not one way to treat anyone. So I would just make sure that you ask them questions.
Okay, so I think we have time for about one more question, so I'll go ahead and try to get to So Marianne hasn't asked a question yet, so I'll go to hers. Does this SNP occur more frequently with females?
No, not that I have seen. Um, I haven't looked into that though.
Mm-hmm.
So I think, we all have these SNPs. And we're passed on through our parents, and we get one of each from each parent. So if it was more down the female line, then we would all probably just be heterozygous rather than homozygous. And there's a lot of people that are both heterozygous and homozygous. So it's obviously passed down to both lines. I think that women may have more problems because of the pregnancy and the fertility and the menstruation, because we need that folate and we need the B12s to continually turn over our cells for pregnancy and to create red blood cells. And so during those times, during pregnancy and menstruation and those times, women, this will play an important role with women. Like I find during pregnancy, it's one of the first times that MTHFR is expressing, usually in miscarriages, and that's kind of the first time, or maybe neural tube defects, or and you can have MTHFR and not express it until a long time later, but that's clinically what I'm seeing is that's the first time we're really seeing problems with it.
Interesting. Okay, well, this is a quick question, so I'll read Natalia's question real quick. She would like to know, can kids be tested?
Yes. If you go to the 23andMe, they have a saliva kit, and there's an extra helper. I forget what it's called right now because I don't deal with children, but they have a extra saliva thing that helps you collect the saliva, because it is a saliva test. There are blood tests, but again, they're pretty expensive. They range about usually around $500 to $1,000 for the same test.
Awesome. Well, thank you so much, Coleen. This was such an interesting and informative talk, and I think everybody really got a lot out of it. So thank you so much for joining us today.
Well, thank you, Renee, and I appreciate you having me.
Yeah, it was a great talk. And thank you, everybody, for attending our webinar. And just keep an eye out for our webinar recording, which will go out within a few days. And also, if you enjoyed our webinar, you can also look into ACHS's online courses in holistic nutrition. We offer single courses, diplomas, certificates, associates, and master's degrees, so there's something for everyone. And also, if you'll be at the NANP conference in St. Paul, both Coleen from Harvesting Nutrition and ACHS will be there to talk all things nutrition with you, so be sure to get in touch with us and let us know if you're going to be there so we can catch up with you in St. Paul. And again, thank you everyone for your great questions and for attending this free ACHS webinar. And please make sure to subscribe to our email list so you can receive future announcements about our Master Lecture Series and any free webinars we may be offering. So thank you again, Coleen, and thanks everyone, and have a great day.
Thank you.